So, can you start by telling everyone your name, your institution, and your role?
I'm Zheng-Yi Chen from Mass Eye and Ear and the Harvard Medical School, and I'm a PI on this project.
Great. And that's a great segue. Can you describe what your current Phase 2 SCGE research project is in a couple sentences?
Our SCGE project is ready to use genome editing to develop product for two genetic deafness. That is, we want to use the same delivery tool, same editor, and same injection route. All the differences between two guide RNA, but then that can be used to target two different types genetic deafness.
And then, what specifically do you work on in the research project? Like, what's your role specifically?
So this project is really develop editing to target multiple in a year, disease, which is genetic deafness. My role, our group, there are 5 groups within this project. My group is ready to test the editing efficacy in transgenic animal model by escalating dose. So we use AAV-mediated genome editing to target 2 mutations in 2 transgenic animal. We will use a different dosage to understand how safe it is, the maximum safety dose to treat genetic deafness in the animal model.
Great. And what's your favorite thing about this research that you're working on?
So my favorite thing is really about this is a project that will enable us to move our work from research and preclinical study all the way towards clinical trial. We have been working on genome editing for genetic deafness for many years and now have a great success to rescue hearing genetic mouse model over the years for different type of genetic deafness. But the biggest question for us has always been, how likely is it going to work in human? So this is a absolute favorite thing is by the end of the study, we'll have a really clear understanding how well this approach will likely work in humans. And so if everything goes well as planned, we will be able to conduct clinical trial.
And then, what's something interesting or cool that you found in your research that maybe you weren't expecting?
Actually, working with David Liu's group, we are the first developer editing and technology to treat genetic hearing loss. When we start the project, technology is still really new. And at that time, we even wasn't sure we could deliver everything in a year, let alone treating disease. And we did that. We showed we could deliver editing region into a year, and we can actually rescue hearing from genetic mouse model. Then the big question for us is, will this potentially work in human? How do we know? Because actually, there hasn't been any type of treatment for hearing loss, for any type of hearing loss, let alone genetic hearing loss. So the only treatment for any type of hearing loss is a cochlear implant for profound hearing loss patient or hearing aid, or device-based. There's no real treatment. So that's a big unknown for us, whether this one will work in human at all. Is there any chance? We are very lucky in that sense, because recently we have conducted a clinical trial by gene therapy, which is based on the work in the animal model, mouse model. And we showed by gene therapy, we can restore hearing in human. We have done all the animal work by genome editing. So we think we are in the exactly same spot. That is, if we can prove it's efficient and safe, we will be able to do clinical trial and we will see the good outcome. I think to move the stage from complete research to directly help patients, that's the coolest thing I can ever imagine.
And now going a little more broadly than your SCGE project, what's an advancement that you think will happen in genome editing or gene therapy in the next 10 years or so?
So genome editing has been so powerful and progressing so fast. But I have a dream, and I think everybody else in the field is the same. Actually, two. One is we want to be able to do like a one standard approach, and that can be used for all genes. That is, we can insert a large piece of DNA, for example, can replace 40 genes in one go. So at the moment, all this editing approach is a sequence-based, mutation by mutation. If a gene has 100 mutations, you literally have the potential to develop 100 different editing treatment, which is very impractical. We're hoping, I know the field is working on that, is we can have a way to insert the whole piece of DNA can totally replace the 40 gene or DNA segment. So as a result, we can use one standard approach to, to target all these potential mutation. The other one, we need to see real progress in the field. I like to see is delivery. At the moment, the bottleneck is also the delivery. If you want to deliver right cell types without toxicity in other cell types, is still a major issue. So we want to see that's the area we can, the field can work on. Hopefully in 10 years, there's a very new and novel delivery approach where you can deliver whatever material to the cell type, the tissue, organ you want, but not to unrelated parts of the body.
Then we're kind of switching gears a little bit. How do you and your team engage with patients or patient advocacy groups in your research?
We talk to patients and, like I got an email from patient daily. And what we want to do for this project, part of the study is on patient natural history. We will, talk to patients to gauge their interest, and of course, to see if this is something they want. We only want to work with the patient if they have a real need, a real desire to be treated down the road. And we have also been talking to some patient groups, and as I said, for vast majority of them, the feedback has been positive. They want to have a treatment for many, for themselves, for patients, for children especially. And there are some incidents where, because of so-called deaf culture, and they want to maintain their deaf culture. So in that case, they actually don't see the treatment as advancement, but as an interference to the lifestyle. So we are very careful with a situation like that. We don't want to be sort of consider intruding their life, and we want to make sure the desire respected. We only work with people, patient group, and they only want this kind of treatment.
That's great. Thank you for sharing that. And then how do you and your team consider health equity in your research?
Yeah, so the one thing is we are targeting ultra rare disease only affecting certain ethnic groups. So, but potentially that can be found in many other groups. So we're always trying to, if we design the experiment, we're talking to patient population, we always want to see one be inclusive, either from genetic ethnic background or male, female, all those things. We want to be all inclusive. And this is the same when done eventually when we develop treatment and we want to clinical trial. We want to be the opportunity to be equitable to all those patients. And so they all feel they have stake in that, they have equal treatment, they have equal chance to be enrolled, recruited, should they choose to participate.
Is there anything else that you would like to say about your research or the SCGE consortium more broadly?
I think every group within SCGE programs work on something really fascinating, unique, has a lot of health benefits to patients. But we're also dealing with similar problem, that is how to navigate, the same issue everyone's dealing, editing efficiency, off target, safety issue, how to navigate the interaction with the FDA. And so in that sense, whatever we learn, we want to people really share within the group, because all will help each other. Almost every one of us will go through similar process. If someone ahead of us have learned something very important, say FDA really focus on this one. So when you design whatever, you should come up with this idea to address that issue or any other related information. I think to be able to share among the group that will within program that really accelerate the whole consortium, not only for one project, for many different projects. So I think that's really very, very important. Actually, one of the most important benefit, I think, from this consortium. The other one is we are basically trying to find a path where to streamline the process with FDA, because for us, everything is the same. The only difference is the guide RNA. And yet the diseases we treat can be different. So we want to be able to identify whether there is such a path, we can streamline the process. And if we can do that, then I can clearly see it will really accelerate the many editing treatment for deafness. But in the meantime, I think this idea can be used for many other diseases as well. So in a sense, we are super excited about the project. I think it will benefit greatly and also be able to contribute. Okay, great. Thank you so much.